All right. Good morning, good afternoon, and good evening, everyone. Welcome to today's webinar, Mastering the Basics of Electroporation and Beyond, Optimizing Your Parameters with the Enhanced Neon NxT System. My name is Kevin Pridham. I am a field application scientist with Thermo Fisher, and I will be your moderator for today's event. Today's educational webinar is presented by LabRoots, brought to you by Thermo Fisher Scientific. To learn more about Thermo Fisher, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speakers, Jacquelyn McClenny, Product Manager, and Shahan Molla, the R&D Lead. For complete biographies on our speakers, please visit the Biography tab on the top of your screen. Jacquelyn and Shahan, take it away.
Thank you for that kind introduction. Today, we're going to be reviewing the basics of electroporation and the Neon NxT system, as well as streamlined optimization for large plasmid delivery, leveraging the Neon NxT 8-channel pipette. And then we'll wrap up with some Q&A. So first off, I will start with the basics of electroporation and the Neon NxT system. Transfection is the process of introducing foreign genetic material, such as DNA, RNA, or proteins, into eukaryotic cells to manipulate their gene expression. Chemical transfection utilizes chemical agents, such as lipids or polymers, to facilitate the entry of genetic material into cells. Meanwhile, virally mediated transfection utilizes viral vectors, such as adenoviruses, lentiviruses, and retroviruses, to deliver genetic material into host cells. Finally, electroporation is an example of physical transfection that involves applying an electric field to create temporary pores into the cell membrane, allowing for the entry of genetic material. There are several advantages of electroporation over lipid-based transfection. These typically include higher efficiency and versatility to transfect some hard-to-transfect cell types, such as primary cells and stem cells, for various downstream applications. Additionally, electroporation is generally a quicker process than lipid-based transfection and require no lipid-based transfection reagents. There are 3 key steps in the electroporation process. Before electroporation, the cell membrane is intact and impermeable to large molecules such as DNA or RNA. The cells are typically suspended in an isotonic buffer, maintaining their physiological conditions. Second, when an electric field is applied to the cells, it induces a transient destabilization of the cell membrane, creating temporary pores in the phospholipid bilayer of the membrane to allow the entry of genetic material. Following the application of the electric field, the cell membrane undergoes a process of resealing. The temporary pores created during electroporation close as the membrane recovers its integrity. Understanding these 3 stages is vital for optimizing electroporation protocols. Successful electroporation relies on a delicate balance of multiple factors, a thoughtful optimization of pulse voltage, pulse width, pulse number, cell density, cell health, and cell cycle, along with the considerations for payload characteristic and buffer types, is paramount. All variables that can be manipulated to optimize electroporation performance. We are going to learn more about optimizing these parameters later on in the presentation. At Thermo Fisher, we utilize our Neon NxT Electroporation System for applications requiring physical transfection. The Neon NxT system streamlines the optimization process for fast and efficient delivery via simple, flexible, and customizable protocols using our electroporation tip technology, where the electroporation takes place inside of the pipette tip. This technology is featured in over 12,000 peer-reviewed publications. The system allows researchers to optimize their electroporation parameters to efficiently transfect hard-to-transfect cell types, but also includes over 400 pre-optimized protocols and over 150 cell lines on the instrument itself. The electroporation protocol with the Neon NxT is very simple. First, we start by preparing the pipette station for transfection by adding the electrolytic buffer. Next, after adjusting cell count and concentration, the payload is added to the cells. Then we simply load the Neon NxT tip into the Neon NxT pipette and aspirate the cells with the payload into the Neon NxT pipette tip. Then we dock the Neon NxT pipette into the station. At this time, we select the desired voltage, pulse width, and pulse number of pulses on the Neon NxT instrument screen. Then we press electroporate. The instrument will indicate when the electric field is applied, and then finally the pipette can be removed from the station with the now electroporated cells and dispensed directly into the proper vessel containing media. Looking at the electroporation profile, more specifically, the electric field strength is dedicated by the voltage and width of electrical pulses. Multiple pulses can also be used as a strategy to improve the efficiency, viability, and control of the process, making it more versatile and effective technique for introducing molecules into a variety of cell types. Aside from optimizing the electroporation profile and electric pulses, selecting the right electroporation buffer for your specific application is crucial. The Neon NxT kit contains one electrolytic buffer that can be used for all applications and 3 different resuspension buffers that you can choose from to optimize for different applications. Our buffer is optimized for efficient and gentle electroporation of mammalian cells and is ideal for transfecting a wide range of cell types, including primary cells and stem cells. T buffer, though, is designed for hard-to-transfect cell types, specifically those that need higher voltage or are more heat-sensitive. Finally, the Neon NxT electroporation kits also contain genome editing buffer, GE buffer, which can enhance genome editing efficiencies, especially for knock-in efficiencies. We have observed this for several cell types, including activated primary T cells. Here, you can see 3 different sets of data where we looked into all of the buffers to identify their key applications and key strengths for each of these cell types. So on the far left-hand side, you can see for primary activated T cells for knock-in and knock-out work, specifically for knock-in, the genome editing buffer produced the highest editing efficiency. Meanwhile, in the middle, we took a look at primary resting T cells, a very challenging cell type to transfect, with both mRNA and plasmid delivery. You'll notice here that T buffer was the right choice for resting T cells. Whereas lastly, with Jurkat cells, we took a look at 2 different plasmid sizes. And you'll notice here that T buffer did better with the large plasmid as well. So this just goes to show the importance of selecting the right buffer for the right application, and optimizing is very, very critical here. Additionally, we wanted to showcase the exceptional performance that you can see with genome editing buffer. Here we took a look at Jurkat K562 and activated primary T cells for knock-in work compared to our buffer. And you'll see there is a significant increase here for the genome editing buffer. It is also important to note how electroporation parameters affect transfection efficiency and cell viability. For voltage, the voltage applied during electroporation determines the strength of the electric field and the extent of the membrane disruption. Higher voltages can cause irreversible membrane damage, while lower voltages may not produce enough disruption to allow efficient uptake of your payload. Pulse width or duration of the electrical pulse also affects the efficiency of electroporation. Longer pulses can produce more efficient uptake, but it can also cause more cell damage, while shorter pulses may be less efficient but cause less damage. Finally, pulse number. The number of electrical pulses applied during electroporation affects the amount of material that can be introduced into the cells. Multiple pulses can increase the uptake of efficiency, but too many pulses or prolonged pulsing can cause significant cell damage and cell death. And you'll see here these various trends across the increasing voltage, pulse width, and pulse number, and how they affect both transfection efficiency and cell viability. Figuring out where to start your optimization can be a daunting task, but we've made it simpler by utilizing tools like the Neon NxT 24-well optimization protocol. This is a pre-programmed optimization protocol where 24 conditions with various voltage, pulse width, and pulse number combinations are preloaded onto the instrument, both as individual programs and in a plate format. This provides a great starting point to optimize electroporation for any new cell type, where you can just start to establish the correct voltage, pulse width, and number for transfection efficiency of your particular cell type and payload. And from there, you can continue to optimize and narrow down to find the exact parameters that's going to be best for your application. The results of a 24-well optimization protocol can help you choose a suitable protocol that balances efficiency and recovery of your cells. So the graph above shows the relationship between electroporation knockout efficiency versus cell recovery and the total viable edited cells for HSCs. As you can see, with higher knockout efficiency, cell viability and recovery decrease. So you can choose between the different programs that result in either a higher percentage of transfected cells or a higher total number of edited cells, depending on your applications and needs. As a note, higher viability indicates healthier cells that are more likely to exhibit normal physiological functions, even though initial recovery may be lower. The cells exhibit a robust ability to recover over time, reaching similar viabilities. Aside from the 24-well optimization protocol, you can also use Transfection Lab, a digital experiment design app, to design your own electroporation experiment in different plate formats. After setting the parameters of your experiment, Transfection Lab will automatically generate a step-by-step experiment guide, including all the calculations done for you, tailored to your experiment. The protocols and plates on Transfection Lab can also be imported directly onto the Neon NxT utilizing Instrument Connect. For more information about Transfection Lab and Thermo Fisher Connect, please check out the resources below, as we do have a link for that there. We did also want to share some very exciting news. Coming soon to a lab near you will be the Neon NxT 8-channel pipette. The Neon NxT 8-channel pipette and station will be an addition to the Neon NxT 1-channel system that has already been out and will now complete the system, if you will. So the 8-channel pipette utilizes the same tip technology with the same proven performance that's been featured in over 12,000 peer-reviewed publications. It also allows for that same flexibility of the parameters, but we're actually going to take it a step further. So inside of the buffer tube, each tip is isolated on the 8-channel. So you can allow for each tip to be a different electroporation parameter set, buffer, delivery payload, or cell type, offering you full flexibility over the system. With the 8-channel pipette, you also can electroporate 96 samples in less than 15 minutes with the plate mode function. And then, as it says, one system, right? So it is one system and one set of consumables for the entire instrument. So there's no need for an additional device. It is all in one at this point. I'm very excited now to pass this off to Shahan to talk more about the different application work we've been doing with the 8-channel pipette.
Hello everyone, I'm Shahan Molla. I'm with Thermo Fisher R&D. In this section, I'm going to present some optimization data with Neon NxT 8-channel. So in this study, we aim to optimize various electroporation conditions to establish a comprehensive guideline for maximizing electroporation efficiency using Neon NxT 8-channel pipette. Specifically, we optimized parameters including electroporation programs, resuspension buffer type, cell concentration, payload concentration, and post-electroporation recovery time for the successful transfection of plasmid DNA into Jacquelyn cells. In addition, we performed a DOE study to explore optimize these variables simultaneously. A notable advantage of Neon NxT system with the 8-channel pipette is its flexibility in applying different programs to different wells. This feature simplifies the optimization of multiple programs with minimal effort. So in this study, we aim to evaluate the optimal electroporation program and resuspension buffer for 5 kb GFP plasmid transfection into Jacquelyn cells. Percentage GFP positive cells obtained from 24 hours post-EP flow cytometry analysis was plotted against the total number of viable transfected cells evaluated from the event count of the flow cytometry analysis. Navy blue data points represents the results obtained with R buffer, while yellow data points represent the results obtained with T buffer. Each data point in this graph represents a specific electroporation program. A total 48 different electroporation programs were used in this study. In the first column, we have Neon 24 optimization program that comes with the instrument as defaults. In addition, we created 24 more higher voltage programs with similar pulse width, with smaller pulse width and pulse number. The red circles in the graph indicate the data points that represents the best performing condition characterized by delicate balance between the higher percentage of GFP positive cells and higher total viable number of transfected cells. All blue points at the top right of the plot indicates that R buffer performed best for the 5 kb GFP plasmid transfection into Jacquelyn cells. In R buffer and with the programs indicated by the red boxes, we achieved up to 95% transfection efficiency. So we have performed a similar study for a larger 11.5 kb plasmid delivery into Jacquelyn cells. In this case, all yellow points at the top right corner of the plot indicates that T buffer performed best for the delivery of larger plasmid into the Jacquelyn cells. In T buffer and with the programs indicated by the red boxes, we achieved up to 85% transfection efficiency. So we selected 7 best performing programs characterized by delicate balance between transfection efficiency and viability, as marked in the red boxes here for the next step of optimization. So in this study, we wanted to assess the impact of cell concentration on the electroporation performance. So we transfected Jacquelyn cells with 11 kb plasmid using 3 different concentration of cells. So we used 5 million cells per ml, 20 million cells per ml, and 50 million cells per ml. This box plot shows the percentage GFP positive cells and cell viability obtained from flow cytometry analysis conducted 24 hours post-electroporation against the EP programs. The figure provides clear evidence that different cell concentration affect electroporation performance. While the impact on percentage transfection efficiency was minimal, 5 million cells per ml slightly outperformed other concentration across all 7 programs. However, in terms of cell viability, 50 million cells per ml exhibited significantly better performance compared to other concentration across all 7 programs. Based on these findings, we have identified 50 million cells per ml as the optimal cell concentration for the further optimization of electroporation conditions in the transfection of 11 kb plasmid into Jacquet cells. So in this study, cells were transfected with 11 kb plasmid using 3 different payload concentrations. The box plot shows the percentage GFP positive cells and cell viability obtained from flow cytometry analysis conducted 24-hour post-electroporation against the EP program. The figure provides compelling evidence of impact of different payload concentration on electroporation performance. For the plasmid concentration of 0.2 milligram per ml, the percentage of GFP positive cells was significantly higher compared to other concentrations, while the cell viability was significantly poorer. The difference in cell viability between 0.05 milligram per ml and 0.1 milligram per ml was minimal. The 0.1 mg per ml plasmid concentration exhibited significantly higher transfection efficiency compared to 0.05 milligram per ml. Based on these findings, we have identified 0.1 milligram per ml as the optimal payload concentration for the further optimization of electroporation conditions in the transfection of 11 kb plasmid into Jacquet cells. So in this section, we have optimized recovery time for the delivery of larger 11 kb plasmid into the Jacquet cells. In literature, it has been suggested that post-EP recovery time improves the EP performance of larger plasmid. So we aim to optimize post-electroporation recovery time using 3 different recovery time. Cells were added into culture plate without media and incubated at room temperature before adding media for certain period of time. We evaluated its impact on transfection efficiency and viability. For the recovery time of 30 minutes, the percentage of GFP positive cells was significantly higher compared to other recovery times, while cell viability was poorer. While the difference in transfection efficiency between no recovery time and 15-minute recovery time was minimal, the 15-minute recovery time exhibited higher viability compared to no recovery time. Based on these findings, we have identified 15 minutes recovery time improved electroporation performance for larger plasmid transfection. In the previous sections, we performed step-by-step EP parameter optimization while keeping some parameters constant. That way, we cannot determine the interdependencies between the factors. Optimization can be performed for all these factors all at once using a DOE study. Here in this DOE study, we performed total 192 electroporation reactions. That means 296 well plate. With Neon NxT 8-channel, it can be done within 30 to 40 minutes. Experiment was performed using 2 different resuspension buffer cell concentration ranging from 5 million to 50 million cells per ml, payload concentration ranging from 0.05 milligram per ml to 0.2 milligram per ml, and a total 48 electroporation programs were used. The predictive profile from the DOE study is shown here with maximized desirability. From the DOE predictive model, we identified optimal conditions for 5 kb plasmid delivery into Jurkat cells. We recognize that R buffer 50 million cells per ml cell concentration, 0.2 milligram per ml payload concentration, and the EP program 2400 volt 5 millisecond and 2 pulses are the most optimal conditions for the delivery of 5 kb plasmid into Jurkat cells. To summarize, the Neon NxT 8-channel pipette significantly streamlined optimization to identify the optimal conditions through individual variable optimization or design of experiments. When considering electroporation, it is important to tailor the approach based on specific needs. This may involve optimizing for performance such as transfection efficiency, cell viability, the delicate balance between the two, or achieving a higher total number of transfected cells. Different conditions can affect electroporation performance. However, electroporation program remains the most crucial factor with the greatest impact on the performance. With the optimal electroporation conditions, 11 kb plasmid was delivered into Jurkat cells with up to 80% transfection efficiency, and 5 kb plasmid was delivered to Jurkat cells with up to 95% transfection efficiency. With that, if there is any questions, I'm ready to answer.
All right. Thank you, Jacquelyn and Shahan, for your great presentation. We will now begin the live Q&A portion of the webinar. We have a good number of questions that have already come in, but if you have not already submitted any questions, please feel free to do so now. Just simply click on the Ask a Question box located on the far left side of your screen, and we will answer as many of those questions as we have time for. So now let's go ahead and get started, and let me load up the questions. And let's see here. Here's a good question. So when is the Neon NxT 8-channel pipette expected to be available? And if I already have a Neon NxT system, do I need to upgrade my whole system, or how can I buy the pipette and the station?
Sure, I'm happy to take that, Kevin. So we are so looking forward to it launching next month, actually. We are expecting it around mid-October. So there's been a lot of development there, and it's finally here, so we're very excited. In terms of if you already have a one-channel system, we will be offering an upgrade package, which includes not only the pipette, but the station, because of course you'll need that. As well as there will be some software features that will be required as a software upgrade for the 8-channel specifically. So that will be included in this package, as well as a service contract to make sure that you are fully protected. So that will be sold all together, and that will be available again mid to late October.
All right, great. We have a question here. This might be a good one for you, Shahan. Have delivery efficiencies been compared between the single channel and the 8-channel device? And are they similar?
Yes, it has been compared using at least 5 different cell types. And we have seen comparable result across all different cell types between 8-channel and 1-channel. Jacquelyn, would you like to add something here?
Yeah, just to continue on that note, not only did we do a comparison between 1-channel and 8-channel, but we also wanted to make sure that things were consistent from a channel to channel perspective, as well as a run to run perspective. And everything has been extremely comparable. Shahan, not sure if you have any details to highlight there, but everything that we've seen, we say it's the same technology. We know it is from an engineering perspective, but biologically, we also see it in the performance.
Okay. Another question we have is there a particular reason why both the 5 kb and the 11.5 kb plasmid was utilized for this optimization, and what key differences were discovered for optimal delivery?
Yes, I can answer this question. So in this study, we wanted to have a representative size of payloads. In the previous studies, it has been reported that with larger payload size, the electroporation efficiency in terms of viability is poorer, as well as transfection efficiency is lower. So we wanted to have a larger payload size to give our customers an idea how to optimize electroporation parameters for a larger payload, as well as we wanted to see how that parameters can be different as compared to a smaller payload, which is here we use 5 kb. So in this study, we have used 11 kb plasmid, and the key difference we observed in terms of the resuspension buffer. So for the 5 kb plasmid, we observed the best performance with resuspension buffer R, while for the larger plasmid, we observed the best performance with the resuspension buffer 11.5 kb resuspension buffer T. And the other differences that we observe is with the high voltage program. Neon NxT system comes with the 24 optimized programs. In this study, we have utilized 24 additional higher voltage program, and we have seen a significantly higher performance for the delivery of larger plasmid with those higher voltage programs. So overall, in this study, we have reported total guideline how a user should step by step should utilize Neon NxT for the optimization of larger plasmid.
Okay. We have some other questions coming in. Let's see. Kind of going along with the optimization protocol, there's a question here about how essentially did you settle on those 24 high voltage protocols, and why were they utilized for this optimization process?
As you know, one of the greatest advantage of Neon NxT system is you can customize the electroporation parameters as you need. And the other advantage of 8-channel system is you can utilize a different electroporation program on different channels. um, so it makes easier to optimize your electroporation parameters overall. um, for in this case, we have used total 48 programs. Out of this 48 program, 24 programs comes with the Neon NxT, that is as default, and we have used 24 additional higher voltage program. And how we selected those program is like for these programs, you can utilize any numbers between 100 to 2500 volt. In terms of pulse width, you can utilize any number between 1 to 100, and pulse number wise, you can utilize any number between 1 to 10. However, you have to keep in mind that the instrument can take up to certain amount of energy. If you go beyond this energy limit, the instrument will show an error message. So if you want to use higher voltage program with higher number of pulse width and pulse number, the instrument will lead to an error. So in that case, what you can do is, like with the higher voltage program, you can lower your pulse number and pulse width, and that would allow you to utilize higher voltage programs for electroporation. So what we have done for this additional 24 program is like we took the voltage from 1800 to 2500 and kept the pulse width within 15, and then pulse number within 3. So that way we have utilized this higher voltage program for obtaining higher efficiencies.
And just
Okay.
to add to that, Shahan, sorry, Kevin, the reason why the instrument shows that error message is those extreme energies can be dangerous to cell types, to cells. And so, in order to prevent arcing from happening, we have done a lot of testing to make sure that we are within that range. Again, we're trying to give optimal flexibility while also protecting cells, and that's why that the instrument will give such a message. In addition to that, Shahan, you probably spoke to this as well, but just to clarify too, these high voltage programs are really ideal with the T buffer. And so that's what we wanted to really understand is how did T buffer combined with these high voltage programs do for large payload, for larger payload size that may need additional oomph, if you will, to get in there. So that's what we wanted to look at.
All right. We have another question here. Is it possible to use the Neon NxT to transport microRNA into extracellular vesicles rather than cells?
We have seen in the literature that people have used neon for the delivery of mRNA into exosomes, but for our internal in R&D have not produced any data for the delivery of mRNA into exosomes.
Let's see here. What are the differences between the resuspension buffers, and do any of them contain components with animal origin?
So the only one that contains an animal origin component is going to be buffer T. Like we said, that is for higher voltage or lower conductivity, heat sensitive cells. We found that particular cells, such as naive T cells or resting T cells, excuse me, do prefer that buffer. In addition to that, our buffer, as we've discussed, is kind of your bread and butter. It's what works for most cell types, for most applications, most payloads. And then we already discussed T buffer. The final buffer is the genome editing buffer, which we released last year around July timeframe. And for it is specialized for genome editing, particularly for knock-in efficiencies. So those are what those three buffers are most used for. But again, as you've seen through this entire presentation, we highly recommend optimizing between the three for your specific use, because you may see one result with one buffer just based on these guidelines. They're general, they are helpful, but by optimizing between the three, you may see better results with a different buffer.
All right. Do Neon NxT tip kits and buffers work with the older Neon system or vice versa?
So the buffers are identical in terms of RNT buffer. GE buffer, like we said, we added in last July specifically into the Neon NxT kits. The tips are not compatible between the two. We did re-engineer the tips for clip tip technology in order to make things easier in terms of attaching and detaching, and also making sure that there's a better seal there on the tips themselves. So it also improved ergonomics quite a bit. So because of that, the tips and the tubes are not compatible.
Okay. We have a question here that I actually might be able to take. It is in regards to any suggestions for utilizing the system to transfect bacterial cells. So there are a good number of papers out there in the literature that do show folks using the Neon system to electroporate bacterial cells. It is a cell type that we do not technically support from our R&D side. However, it has been demonstrated that the device works for bacteria. I would suggest if you wanted to try using it for that situation, the best situation would be to have a separate pipette and pipette station if you also do electroporation of mammalian cells. That's a really good way to help prevent any risk for cross-contamination between your devices. And if you're not able to have two devices, I would definitely make sure you thoroughly clean the unit and the pipette between uses, between species of cells. But definitely take a look out there in the literature and you will find suggested protocols that you could try.
Thank you, Kevin. Yep, the instrument itself was not designed for bacteria. It was optimized and designed for mammalian cells, but as you said, we have seen it in the literature. So great points.
That's one of the things I love about the Neon system is it's an open book and you can go in the lab and try stuff out. Another question here is, how does the Neon NxT system differ from the original Neon transfection system?
So many things. Oh my goodness. We really tried to redesign it. The original Neon system is quite old at this point. We did choose to discontinue the instrument itself last year for that reason once we had the Neon NxT come out. But it is the instrument itself, the box, if you will, with the screen is significantly smaller. You don't even have to raise the sash of your biosafety cabinet to get it in there now. It just slides right in, which is great. The screen in the user interface has been completely redesigned, brought into this decade and everything else. And so there's now plate mode, which helps speed up your experiments. It really enables you to plan things out ahead of time. In addition to that, we already talked about Transfection Lab. That was also a tool that we released with Neon NxT, and that allows for that full remote experiment planning where all you had to do is send over the plate to the instrument itself and import it there, and then you have a step-by-step guide. So that goes hand in hand. The ergonomics are a lot better with the pipette. And then again, the genome editing buffer, I think, is a great added benefit. So a lot of improvements, but we are super excited for this 8-channel pipette to come next month, as that is going to really take us into the future and enable a lot faster electroporation while still maintaining that maximum flexibility.
Okay, so let's see. I think the last question will be for Shahan. It's regarding the design of experiment predictive profiling slide that we had in the talk. Have you used the predictive profiling to look at the voltage, the pulse width, and the number of pulse parameters independently to predict which one will influence the percent GFP or viability the most?
Yes, that's a great question. In this study, we have taken EP parameters as a whole. We have not looked at them separately, but it would be a great DOE study to look at those factors separately and design an experiment to find out best parameters for a particular delivery.
Okay, great. Let me see here. Just to give a couple more seconds for any last minute questions to come in, I'll open the floor to both you, Shahan and Jacquelyn, if you have any final comments or things you would like to mention before we close it out.
Honestly, just thank you everyone for attending. Like we said, we're really looking forward to 8-channel coming out next month. We hope that it's going to speed things up for everyone, again, while maintaining that flexibility. And the other thing to note is, Shahan, actually, question for you, if you can refresh my memory, how many 96 plates were you doing in one sitting for this experiment with the 8-channel?
I think the largest experiment that I did was 296 well plate for this DOE study. And you can imagine 296 well plate or 192 reactions. So it was done within 35 minutes, I believe.
Wow.
So just think about it. If you are doing one channel electroporation and 192 reaction, it would take you hours to complete. But we were able to complete the whole experiment within 35 minutes. So it saves our time and resources significantly.
That's great to hear. And thank you again for refreshing my memory. I know there were multiple plates and so many experiments done to pull together this data, and it's a wonderful data package. It's great to have support on that large plasmid, as I know this is something that a lot of folks are looking into right now. And so with that, Kevin, anything else from you?
Let me check one more time on our question box, but I think we hit all the questions. So we will go ahead and round this out. So thank you again to Jacquelyn and Shahan for your time today and for your great presentation. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for providing today's educational webcast. Before we go, I would also like to thank those in the audience for joining us today and for their interesting questions. If there are any other questions that come in that we're not able to get to today, we will be able to follow up with you. This webinar will be available on demand. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with any of your colleagues who might have missed today's live event. We also invite you now to partake in a survey on the left side of your screen. We would be grateful if you spent just a couple of minutes answering the questions. It helps us to learn if we have covered all of your expectations, as well as to just improve the experience in upcoming webinars in the future. Until next time, take care, everyone, and goodbye.